Okay, going back from the start, we are done with the drug discovery, which would took up 2 to 5 years or 3 to 6 years. Then, if you have your drug candidate, we proceed to the preclinical development stage, where drugs are tested in vitro and in vivo and in silico. Then, application of IND before the clinical trial. After 30 days, if no problem or no clinical hold issued by the FDA, we may proceed to the clinical trial. We will now cover the different types of clinical trial designs and protocols, as well as the roles of the various stakeholders involved. The objectives of this video are to differentiate various types of clinical trial designs, describe the components of a clinical trial protocol, and discuss the roles of different stakeholders in clinical trials. Let me define with you some key terms we may encounter in the long run for these topics. Cohort study. A cohort study observes a population over time to estimate disease or life event frequency. An example of this is smoking, which in time may lead to lung cancer risk. A cross-sectional study measures prevalence at a single time point, which prevalence refers to the total number or proportion of individuals in a population who have a particular disease or condition at a specific time or over a specified period, meaning the study of how common is the disease. Example, percentage of students with hypertension. Case control study. Compares diseased versus non-diseased groups. Example, we compare the COVID patients versus healthy individuals. Next, randomized clinical trials. Assign eligible participants randomly to treatment groups, meaning participants who meet specified standards with inclusion or exclusion criteria are assigned by chance to different interventions. While non-randomized trials do not. Participants may choose which group they want to be in, or they may be assigned to the groups by the researchers. Clinical trial design is crucial because poor planning can lead to errors, meaning if you have not properly planned the details of the study, you may have errors in the middle, or worst, after you've finished all the study and you have seen that there is an error on how you conducted your study, which may have an erroneous results or unreliable results. Remember the principle. Good statistics cannot fix a badly designed study, meaning even if you go to a very knowledgeable statistician, if the results are not good, therefore your statistician may not have a good output. Technically, your statistics really rely on your data and how good your data are. Therefore, trials must be carefully planned to determine the drug's safety, efficacy, side effects, optimal dosage, meaning the dose that the drug would give the best therapeutic effect, and comparison with standard treatment, meaning you compare it with the established drug already. Clinical trials aim to determine the true effect of an intervention, meaning you study the current drug's effect. Does it really lower the blood pressure or not? So we have methods in clinical trial. Key methods include randomization, assigning participants by chance, blinding, hiding treatment allocation, which we have single blind, only the patient doesn't know which treatment they're receiving, double blind, in which both patient and clinician are unaware of treatment assignments, and open label, meaning all participants know what is being administered. Using a control group for comparison, which in the next slides we will tackle more of this, and ITT, or intent to treat, analyzing participants based on their original assignment. Clinical trials are the human testing phase of a drug, meaning after the preclinical testing, which drug has established safety and effectiveness, because we don't want to test in human if during clinical trial that the drug is proven to be toxic. So going back, clinical trials are procedures in medical research that are conducted to collect safety and efficacy data for health interventions. Safety includes information on adverse drug reactions and effects. So why is it important? We want that the public is informed of the possible interactions and adverse reactions on taking the medication. And most people have other concomitant disease that may or may be not contraindicated with the drug. Clinical research encompasses all scientific approaches to evaluate medical disease. Research, meaning you are investigating with the help of scientific data. This includes prevention, diagnosis, and treatment. Clinical trials are a specific type of clinical research. Let us try to compare the two. Clinical trials involve human testing of experimental interventions, like drugs or experimental, covering prevention, diagnosis, and treatment. And therefore, all clinical trials are clinical research, but not all clinical research are clinical trials.
There are several types of clinical trials, including treatment trials, prevention trials, diagnostic trials, screening trials, and quality of life trials, also known as supportive care trials. Treatment trials test experimental treatments, new drug combinations, or new approaches to surgery or radiation therapy. Their purpose is to evaluate new therapies, such as new antibiotics or cancer chemotherapy combinations.
Next is the prevention trials, which these trials look for better ways to prevent disease in people who have never had the disease or to prevent recurrence, meaning you want to prevent having a disease or prevent a recurring of the disease. Methods include evaluating vaccines, lifestyle changes, vitamins, and minerals. Very best example, having an HPV vaccine to prevent cervical cancer in the future. While diagnostic trials are conducted to find better tests or procedures for diagnosing a particular disease or condition, in short, we want to evaluate a better diagnostic tools. Examples include evaluating new blood biomarkers, AI imaging, or rapid infection tests. Screening trials determine the best way to detect certain diseases or health conditions early. These trials evaluate methods such as mammography, which is used in screening of the breast, which is usually done in women of 40, colonoscopy, which is a gold standard for colorectal cancer, and also blood pressure screening.
Quality of life trials, or supportive care trials, explore ways to improve comfort and the quality of life for individuals with chronic illness. They focus on aspects like pain relief, nausea control, and sleep improvement.
Again, we are done in drug discovery, preclinical development, investigational new drug application, or INDA, and now we are focused on clinical trials. Clinical trials in humans are typically divided into three main phases. Phase 1 tests safety, dosage, and side effects, usually in healthy volunteers. Phase 2 assesses effectiveness and short-term side effects in patients, while Phase 3 confirms effectiveness, monitors side effects, and compares the treatment with standard treatments. Please see the photo indicating the summary and organized 3 phases of clinical trial.
Next, we will look in more detail at the different phases of clinical testing.
Phase 1 focuses on safety and dosage, usually involving 20 to 100 healthy volunteers. Why small? This is the first time that the drug is tested in human, and testing it in a large volume of human is not allowed. This test is conducted in assessing the safety, determining pharmacokinetics, and identifying side effects and tolerability. The trial starts with a low dose and increases gradually. Phase 2, with 100 to 300 patients with target condition already, which evaluates effectiveness and dose ranging, meaning the dose range that is considered safe, effective, and have best therapeutic outcome. Goals include evaluating efficacy, continuing safety monitoring, of course we don't want to continue if the drug is not safe in the long run, and determining the optimal dose range. Now in this phase, it is divided into Phase 2A, which is for initial effectiveness, and Phase 2B, on the other hand, for dose fine-tuning, with very minimal toxicity. Also, drug formulation is refined here, meaning it could be an IV, tablet, capsule, or the proper dosage form that would not affect the drug's potency in administering to the patients before Phase 3. And if promising, or have good report, an end-of-Phase 2 meeting is held with the FDA. Now, Phase 3 involves large-scale confirmatory trials with 300 to over 3,000 diverse patients. The main goals are to confirm safety and effectiveness, evaluate the benefit-risk profile, is the drug more beneficial than the risk, and finalize dosage and formulation that would be made available in the market. The results support the new drug application. Now it is already an NDA and not investigational drug, and it includes Phase 3A and Phase 3B studies. Another application with the FDA is done, which is the NDA, or the new drug application. Phase 3A data should be sufficient for NDA, which in the next slide will be discussed, and Phase 3B, which is an ongoing study of post-NDA. An NDA is submitted to the FDA after successful Phase 1 to 3 clinical trials to gain approval for marketing a new drug, meaning the drug will be made available in the market for purchase. The goal is to prove the drug is safe, effective, and manufactured with high quality.
This graphic illustrates the progression from preclinical studies through the 3 phases of clinical trials to approval, and finally, post-marketing surveillance, or Phase 4. Now let's discuss various types of clinical trial designs, which include parallel, crossover, factorial, and randomized withdrawal approaches.
In a parallel design, subjects are randomized to one of 2 or more arms, each receiving a different treatment. This is the most commonly used design. In short, participants assigned to separate groups. Example, we have Group A assigned for new drug, while Group B assigned for the standard drug, and we compare the results. Clinical studies use different types of controls. First, we have the placebo control, uses an inactive substance to help detect the true effect of the investigational drug. It is double-blinded and minimizes bias, though it has ethical disadvantages. So, when do we use placebo? Is it used in disease with no prior drugs has been established as a standard therapy? Placebo, meaning an inert substance that looks exactly like test drug, but contains no drug.
In a no-treatment control study, subjects are randomly assigned to either the test treatment or no treatment. A significant drawback is that subjects and investigators are not blind to the treatment, which introduces bias. An active control uses a standard or known drug for comparison with the investigational drug. This design is often used when withholding treatment for a serious condition would be unethical.
While dose-response control compares different doses of the investigational drug within the study, and an external control uses data from patients outside the study, such as from previous trials or medical records. A mixed control study uses both placebo, an inert substance that looks exactly like test drug but contains no drug, and an active drug as comparators within the same study. This design allows for simultaneous assessment against both a baseline and a standard treatment. Next design, a crossover design, in which each patient receives both drugs, and the order of administration is randomized. This design is advantageous as each patient serves as their own control, requiring a smaller sample size. Crossover studies assume the effect of the intervention from the first period does not carry over into the second period, meaning the treatment given in the first phase should not continue affecting the patient in the second phase. That is why there is washout period. Internal and external factors must remain constant over time, meaning these are patient-related factors within the individual that can influence the study outcome. Example, the age of patient, organ of the patient, weight of the patient, and others. Requirements also include a chronic stable disease, meaning the disease should be relatively unchanged over time. Full development of effects within the treatment period, meaning the drug must show its effect during the assigned treatment phase. And drug effects being reversible, meaning the treatment effect should disappear after stopping the drug. And adequate washout, meaning the drug should be eliminated after approximately 5 half-lives. Disadvantages of the crossover design include the carryover effect, which means the effect of the first treatment continues into the next treatment period and influences the results. Period effect means changes happen because of time, not because of treatment, meaning in the long run, the disease has progressed on its own and not due to the drug. And difficulty in attributing adverse events, which the patient cannot determine if the side effect is from the drug. This design is also not suitable for acute diseases or vaccine trials, because vaccines usually produce long-lasting or permanent immune effects. This slide sets the stage for a comparison between the parallel and crossover designs. Lower complex, meaning it is easier to manage.
Factorial design involves 2 or more interventions. This approach is beneficial because it allows for the simultaneous study of interactive effects between the different treatments. In a factorial design, patients are often randomized to groups receiving drug A, drug B, both drugs, or neither. Advantages include efficiency and interaction analysis, while disadvantages are complex analysis and interpretation challenges. The randomized withdrawal approach is particularly useful for determining how long a therapy should be continued.
In a randomized withdrawal study, initial responders are randomized to either continue or withdraw therapy. This design is useful for maintenance treatment studies, such as evaluating beta blockers post-MI, meaning after myocardial infarction.
Clinical trial innovations include adaptive design, superiority trials, and non-inferiority trials. These designs offer flexibility and specific objectives in clinical research. An adaptive design permits modifications to the trial after its initiation without undermining its validity. This flexibility can make trials more efficient. A superiority trial aims to show that a new treatment is better than a control, which could be a placebo or a standard treatment. Examples include placebo-controlled and active-controlled efficacy trials.
Non-inferiority trials demonstrate that a new treatment is not worse than the standard treatment by more than a defined margin. These trials often use active control and are suitable when a new drug offers advantages like better tolerability or less dosing.
Thank you for watching this video on clinical trial design.